Hey guys, welcome back to my channel. Today we are going to learn a chapter in biochemistry known as enzymology. Enzymology is nothing but study of enzymes. Okay. First let us see what is an enzyme. An enzyme is a protein, rather than protein you can say a substance which enhances the rate of reaction. Okay, as you all know that. Then coming down to the characteristics of enzymes, almost all enzymes are proteins except ribozymes, right? They are heat labile, they are water soluble, precipitated by protein precipitating agents, 16% weight as nitrogen. Okay, 16% weight of an enzyme is a nitrogen. Then coming to the IUBMB system of classification, this was made because earlier names were confusing. Okay, as per the system, name starts with EC followed by 4 digits. First digit is class. There are 7 classes. Then subclass. Third digit is subgroup. Fourth is number of particular enzyme in the list. Next, coming down to the classes of enzymes. It is very important to remember them in order. Okay, because the class 1 is oxidoreductases. You cannot put transferases here. Okay, and there's a mnemonic which you can remember if you want. I would suggest you, that is other little so this is a mnemonic: oxidoreductases, transferases, hydrolases, lyases, isomerases, ligases, and translocases. Okay, this is a mnemonic. If you want, you can remember it. Then coming to the classes, class 1 is oxidoreductases. They are nothing but enzymes which oxidize one substance, reduce others simultaneously. It means oxidation and reduction, redox reaction, which facilitate redox reaction. Okay, example is alcohol dehydrogenase. And the IUPMB system, according to that, the name will be EC.1.1.1.1 because it belongs to the first class, first subgroup, first subclass and first subgroup, and the number of particular enzyme in the list is 1. Okay, then coming to second class, that is transferases. Transfer one group from one substrate to other except hydrogen, because if hydrogen is transported, it is known as oxidoreductases, right? Yes. Next, coming to hydrolases. Hydrolases, they hydrolyze by adding water and break the bond. So they usually break the bond by adding water. Okay, example acetylcholine esterase. Okay, this for enzyme, see there's acetylcholine here, it breaks that to choline. Okay, choline using water, by adding water to acetylcholine. Then lyases, they also break bonds but by mechanisms other than hydrolysis. Okay, example is aldolase. Then coming to the class 5, that is isomerases. They produce optical, geometric, or positional isomers of substrate. Okay. Then the examples are racemases, epimerases, cis trans isomerases. Right. Then next is ligases. Ligases are nothing but links two substrates together, usually with simultaneous hydrolysis of ATP. Example is acetyl CoA carboxylase. Okay, this was all about the 6 classes which you have already learned in class 11 or 12. 11, sorry, 11, you learned it in class 11. Then coming to the new class, this is a new class introduced relatively very new, that is 2018. Name is translocases. They translocate ions and other molecules from one side of membrane to other side. They are historically classified under active transport because they require energy. Okay, some important example are carnitine, acyl carnitine translocase. They import long chain fatty acid, long chain fatty acids into mitochondria. Okay, that is CACT. Then coming to adenine nucleotide transporter, it imports ADP from cytosol and exports ATP from the mitochondrial matrix. Understood? For this ATP production, you require ADP, right, in the mitochondria. This will be facilitated by this ANT, which imports the ADP from cytosol. Okay? Then coming down to coenzymes. What are coenzymes? Coenzymes come under cofactors. You all know. Coenzymes, they are non-protein part of complex enzymes. See, a enzyme, also known as holoenzyme, complete enzyme also known as holoenzyme. A holoenzyme is a mixture of apoenzyme and the coenzyme or cofactor. Okay, we'll see that in the later part. They are heat stable and are required for enzyme activity. See, as I told you earlier, a holoenzyme is the entire enzyme consisting of the apoenzyme and coenzyme. Apoenzyme is the protein part, okay? And it is divided into two groups. Coenzymes are divided into two groups. The first group of coenzymes, they donate or accept hydrogen atoms or electrons. Okay, they donate or accept hydrogen atoms or electrons. These are the first group of coenzymes. Changes occurred in substrate is counterbalanced by coenzymes, hence also known as co-substrates. So here, let us take the example of this. This is glyceraldehyde 3-phosphate. The enzyme is glyceraldehyde 3-phosphate dehydrogenase. Okay, this gives out 2 hydrogen and 2 electrons. Okay, this is not accepted by this enzyme, it is accepted by this coenzyme, that is NAD plus, nicotinamide adenine dinucleotide, right? Okay, so in reaction, the substrates are counterbalanced by coenzymes rather than the apoenzymes. Okay, you have to remember it is a very important point. See here in 3.1, substrate lactate is oxidized and simultaneously the coenzyme is reduced. Lactate is hydrolyzed and this is reduced. Chalo, examples are NAD plus NADH, NAD plus NADPH, FAD plus FADH2. Next, let us look into one of the coenzyme belonging to the first group, that is nicotinamide adenine dinucleotide NAD+. The coenzyme synthesized by nicotinamide, okay, it is synthesized by nicotinamide, a member of vitamin B complex. Majority of coenzymes are vitamin B complex members, okay, remember this. The structure is nicotinamide, ribose, phosphate, ribose, adenine, okay. Then the reaction here, the reaction in 3.1, okay, is catalyzed by enzyme lactate dehydrogenase, but actual transfer of hydrogen takes place in the coenzyme, that is NAD plus and NADH. Next, coming to second group of coenzymes. Second group of coenzymes, it take part in transferring of groups other than hydrogen, and they also belong to vitamin B complex. Then metalloenzymes. Enzymes require certain metal ions for their activity. Metals are tightly bound to the enzymes. In some cases, enzymes are active without metals, but metal is added, activity enhances, also known as ion activated enzymes. Okay, this was the cofactors. And earlier I had told you, right, what is cofactor? Cofactor is a collective term that includes metal ions and coenzymes, that is non-protein part. Okay. So that's it for today, guys. Okay, in the next video I will discuss the mode of action of enzymes, okay, and the further topics. And if you have any doubt, do comment, okay. And if you require notes, if you want these notes, you can tell me in the comments. I would send you the link of my Telegram, okay. You can message me on Telegram and get these notes, that too for free. Thank you.